Good evening, students. Welcome to yet another session of our MBBT. So it's been like almost 1 month. We've been continuing with this our various sessions of MBBT, and slowly and slowly, initially we were very slow to turn out with the very basics of molecular biology and biochemistry, but eventually we took the speed and understood so many techniques within last 2 weeks. Still we have 2 weeks more, no, 1 week more, I guess. Next week we will gradually will finish the other topics also. It will be about bioinformatics application, how the scientific ethical work we have to work, and facts analysis, chip analysis, blotting techniques, and so on. So at the moment today I want to take you to the world of electrophoresis, how electrophoresis is done and what are various types. Yeah. So it's almost 8 now, we can start, I guess. So students will gradually join. Who's not able to join in, they can watch it later also, the recorded version. So electrophoresis technique, as so far we have done many techniques already in brief, actually not in detail, but in this time we will do something more like from the subjective perspective, from a Wilson and Walker book and all the biochemistry books that are present which teach about the electrophoresis technique, its various types also. So first of all, as we know, electrophoresis is a physical method of analysis which involves separation of compounds that are capable of acquiring electric charge and conducting electrodes. So electrophoresis may be defined as the migration of the charged particles through a solution under the influence of an external electric field. So ions that are suspended between two electrodes tend to travel towards the electrodes that bears opposite charges. So there are two types of electrophoresis, that is zone electrophoresis and moving boundary electrophoresis. The zone electrophoresis includes your 4 types, that is paper electrophoresis, gel electrophoresis, thinly electrophoresis, and cellulose acetate electrophoresis. Yeah, all various types. And the moving boundary electrophoresis includes your capillary electrophoresis, isotachophoresis, isoelectric focusing, and immunoelectrophoresis. Yeah. So first of all, the zone electrophoresis, in this basically your charged particles are moving along your sporting media, and the sporting media that we use could be polyacrylamide, starch gel, cellular acetate membrane, and paper. So components are separated, distributed onto the discrete zone on the sporting media, and the sporting media is saturated with the buffer solution, that is small volume of sample is applied as a narrow band. So in applying this potential difference to the end strip of components, they started to migrate according to their electrophoretic mobility. It has many advantages as it helps in the biochemical investigation. Cost is low, easy to maintain, and small quantity of sample can be analyzed. So general method of operation in electrophoresis, zone electrophoresis, that you saturation of medium with the buffer, then sample application, then electrophoretic separation, then removal of the supporting media. Yeah. Then in the instrumentation, electrophoretic chamber, in the instrumentation we need electrophoretic chamber, electrodes, diffusion barriers, supporting stabilizing media, and so on. Then the paper electrophoresis, what we have a pencil line like this structure here under the 2 glass plates, and our paper is put into the positive side and negative side. Yeah, and then the migration is done over this paper electrophoresis, and this is also of 2 types, horizontal and vertical. Horizontal shapes goes like this, like totally surrounded in the horizontal shape, which will have around the airtight housing with a positive and negative electrode in the two buffer solutions. And the main difference between the vertical paper electrophores is that the filter strip is there. There is a filter paper strip on the top around your buffer solution. So the filter paper such as Watnam number 1, Watnam number 3 could be used, which has a quite good effect, and the separations in this case with the zone electrophoresis under paper electrophoresis, it takes around 12 to 14 hours. It's quite a lot of time if we see. Then comes your continuous electrophoresis, in which also you have a positive and negative end. Your sample is put over the top and they are being separated like this, you know, these sample collections over the zigzag shape, and in the various test tubes you will have your sample collected. This is used for preparative scale purpose. Predetermined sample volume of 0.2 milliliter per minute through the wall device is applied continuously on the center of the paper. So silica gel, powdered glass and an agar gel can be used, and potential difference of 500 volt is applied. So the pure compounds are collected in separated containers and the solvent is evaporated and pure fractions are reused. So gel electrophoresis is brought about through the molecular sieve technique based on the molecular size of the substances. So gel material act as a molecular sieve in it. So gel is a colloidal in this, as 99% is water, and it is important that sporting media is also electrical neutral. So we can use agar, AGAROSE, gel, starch, SULFATE, polyacrylamide gels and so on. And the porous gel act as a sieve by retarding or in some cases completely obstructing the movement of macromolecules while allowing small molecules to migrate freely. So we have discussed that you put your sample over the gel, and then you put your gel first, then you add the comb, and then you put your sample, then get attached to your electrophoretic supply. So this could be also two types, vertical and horizontal, according to its work. We can use two types of gel in it. One is agar and one is AGAROSE. Agar is a mixture of polysaccharides extracted from the seaweeds, whereas this AGAROSE is highly purified uncharged polysaccharides derived from your agar medium. So AGAROSE is a chemically basic disaccharide separating units of 3,6 anhydro-L-galactose. Whereas the AGAROSE gels are fragile, they are usually hydrocolloids and they are held together by the formation of weak hydrogen and hydrophobic bonds. So this is the structure of your AGAROSE. In the solid state they are all open, and then at the 45 degrees Celsius they tend to make this, started to make crisscross structure. Then after some aging they make this very nice polycolloidal structures. But if you again burn it to heat it up to 100 degrees Celsius, they come back to its initial gel and then the solid state again. So this could go back and forth. So now let's see how AGAROSE gel electrophoresis is basically done in the lab, because this is widely used technique in your day-to-day life. You will definitely gonna come across this technique for sure.
This video will take you through the process for loading and running DNA samples on an AGAROSE gel. You will be using a mini subcell to perform AGAROSE gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the AGAROSE gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the samples spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on. Then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
Yeah, that was AGAROSE gel electrophoresis. Now this polyacrylamide gel electrophoresis. So in this it is prepared by polymerizing your acrylamide monomers. This we also have discussed yesterday in this 2D gel electrophoresis, where acrylamide and methyl bisacrylamide, they make a cross connections together these monomers and make a this section, this figure B like structure. So that they are together very tougher than the AGAROSE gel, and they are quite thermostable, strong and relatively chemically inert. And gels are uncharged and prepared in a variety of pore sizes. So proteins are separated on the basis of charge to mass ratio, molecular size, and phenomena is called as molecular sieving. So gels are stable over a wide range of pH and temperature, and gels of different pore size can be formed, and simple and separation speed is good comparatively. Then types of page, this could be native page and denatured page. So the native page are run in the non-denaturing condition so that the analyte natural structure is maintained, where the separation is based upon the charge, size and shape of macromolecules. So useful for separation or purification of mixture of proteins, and this was original mode of electrophoresis. Whereas a denatured page or SDS page, they are based upon the molecular weight of proteins, and the common method for determining the molecular weight of proteins and very useful for checking protein of protein samples. So this we have discussed the page procedure. I will not repeat this since yesterday. So you run your gel over the high voltage power supply and you get different bending under it. The gel is poured vertically between the two glass plates and proteins bands are separated on the basis of your relative molecular weight and visualize with the states. Then the slabs page. This is a polyacrylamide gel which is cast as a thin rectangular slab inside the plastic frame, and this slab is placed vertically on a buffer solution taken in a reservoir. And we can visualize these gels, protein gels, by ethidium boride, silver or commercy blue dye, which may be used for this purpose. If the analyte molecules fluorescence under the UV light, photograph can be taken. We can use this as under the autoradiograph also. Then the page is added to SDS. We also have discussed in detail yesterday that your SDS-POLYACRYLAMIDE gel electrophoresis with the sodium DODECYL sulfate polyacrylamide gel electrophoresis is a technique of a biochemistry, forensic, genetics, molecular biology to separate your proteins according to their electrophoretic mobility. So SDS actually code over the proteins to give them constant charges to mass ratio. So due to the masking of charges of proteins by the large negative charges on SDS, the proteins migrate along the gel in order to the increasing size of the molecular weights. Then comes your sodium DODECYL sulfate, that is your SDS page, and the native protein is unfolded by heating in the presence of your beta mercaptan and SDS. And SDS binds to the protein so that it stays in solution and denatures it. So we can see we have a native protein in its fully folded structure. So we heat it, reductant is done, and then SDS is covering your protein. And this is now your denatured protein bonded over the SDS. And this is very easy to then transfer over your gel. So SDS coated proteins with a large size, middle size and a small size, they're all being separated according to the charge mass ratio. Then a starch could be also be used, suspension of granular starch, which could be used in a buffer to give a colloidal suspension. So the suspension on a cooling set as a semi-solid gel due to the intervening of the branched chains of myelopectin. So in order to avoid swelling and shrinking, petroleum jelly is used. So this vertical and horizontal gel electrophoresis, how it is done, up is the vertical one, this is the horizontal. So here we having this trough is there which having the starch gel and sample. So they will be over this filter paper, and this will be a filter paper connecting strip. Yeah. So this method for application of sample. So sample is mixed gel. So by soaking a bit of filter paper in sample, pressing it into the gel, and detection by staining, direct gravimetry or UV absorption is done. So pulse field gel electrophoresis. So pulse field gel electrophoresis, this is another very famous technique, very highly used, where we have to separate large DNA molecules over a gel matrix in which we are changing this electric field periodically. So they are going up and down and left and right, all directions, 60 degrees Celsius, 90 degrees Celsius. However, the periodic changing of the field directions, various lengths of DNA, are to change at different rates. The voltage is periodically switched among three directions. One of that runs to the central axis of the gel, and two run at the 60 degrees Celsius of it. So like one is running like this, one will be on 60 degrees Celsius. So they keep changing. So this your pulse field gel electrophoresis. Oops, sorry. So they may use for genotyping or genetic fingerprinting, and it is commonly considered gold standard in the epidemiological studies of your pathogenic organisms. So quite useful this technique also. The next technique is your thin layer electrophoresis. This could be carried out in a thin layer silica or Kysel Gur or alumina. They take very less time, gives a good resolution. On the other hand, it could be used for electrophoretic chromatography studies in two-dimensional 2D gels for the study of various proteins and nucleic acid hydrosylates. Then in your zone electrophoresis, last electrophoresis type, that is your cellulose acetate electrophoresis. So this contains 2 to 3 acetyl groups per glucose unit, and its absorption capacity is less than that of paper. and it gives sharper bands, provides a good background for staining glycoproteins. Advantages are no tailing of proteins or hydrophilic materials, available in wide range of particle size and layer thickness, give sharp bands and offer good resolution. High voltage can be applied, which will enhance the solution. But it's a bit expensive, and because of the sulfonic acid and carboxylic group present, they could cause electroosmosis during the electrophoresis. They are being used for the clinical and biological protein samples like albumin and globulin, and could be a good alternative to your paper electrophoresis. Now comes after the zone electrophoresis, moving boundary electrophoresis. So the basic principle that they allow the charged species to migrate in a free moving solution without supporting media. So in this, the instrumentation we always have a U-shaped glass cell of rectangular cross sections with electrodes placed on each of the limbs of the cell, and the sample solution is introduced at the bottom or through the side arm, and the apparatus is placed in the constant temperature bath at 40 degrees Celsius, and detection is done by the measuring refractive index throughout the solution. So this is your initial boundaries, yeah, where you are having these molecules with anode and cathode, and then you running your samples in between them, and they are being separated according to their charge. So this is observed boundaries by optical system. So biological active fractions can be recovered without the use of denaturing agent, and reference method for measuring electrophoretic mobilities, and minute concentration of the samples can be also be determined. Disadvantage that is quite costlier, and elaborate optical systems are required. An application to study homogeneity of macromolecule system and analysis of complex biological mixtures too. Then within this zone moving bounding electrophoresis comes the next part, capillary electrophoresis. This is also one of the famous one. If you want to study about neurodegenerative disorders, Alzheimer, Parkinson, prions, all the brain. So in that case, this capillary electrophoresis is playing a big role. The principle behind is that the charged molecules will migrate towards the opposite pole and separate from each other based on the physical characteristics. So capillary electrophoresis is a technique of performing electrophoresis in buffer-filled narrow bore capillaries, and normally from 25 to 100 millimeter in the internal diameter. So the high voltage that we use typically is 10 to 30 kilovolt is applied, and typically 50 micrometer in a diameter and 0.5 to 1 meter in length is used. So that's the setups that looks like for capillary electrophoresis. You are having a cathode here and anode. Again, in most of the cases in all electrophoresis, you're having this basic principle, and then you have a mixture of positive and negative and neutral protein particles, and then there's also this light source which get attracted towards the photocathode, which further gives data to the computer, and we can have this chart recorder giving absorbance at the 280 nanometer and over the time of various things. So the sample application is done either by high voltage injection or pressure injection. So high voltage injection, tension is applied causing the sample to enter the capillary by combination of ionic attraction and electroosmotic flow. Whereas a pressure injection is a pressure difference is used to drive the sample into the capillary by applying vacuum. Then comes a third one, electroosmotic flow. In this, we are having, in the silicate glass capillaries are containing with the both negatively charged and positively charged counterions. So we are having these both, this glass capillary, and then we give this electroosmotic flow with a positive and negative end. So over the negative glass plate, your positive things will be attached, and once they got saturated, so in between we will have plus and minus only. So this will be this flow of positive and negative counter ions. This will be called as your electroosmotic flow, which will flow from positive to negative. It will be in between. So on this basis, we can separate our tubes and we put them over the capillary tube, and then hence we get the data acquisition and find the results later on. Then there's an isotachophoresis. I hope you have never heard these terms before, because they are very like kept unique within the electrophoresis. So I hope you are learning some new techniques today within the electrophoresis itself. So this is the technique of isotachophoresis depend on the development of potential gradient. So basically in this also it's the same principle as your moving boundary electrophoresis. A leading electrolyte with high mobility than analytes and a trailing electrolyte with a low mobility are used. The solution in which the separation takes place is normally an aqueous medium which contains sucrose to provide a higher density to the solution. So where the separation by isoelectric FOCSING depends on the existence of pH gradient in the system, the techniques of isotachophoresis depends on the development of the potential gradient. So here we can have three molecules A, B, C over the 10 seconds with the different concentrations are being separated here in this isotachophoresis. So in this example we can see three particles classes with different charges are being separated and pre-concentrated by the electrophoresis. So after the separation is concluded, all the particles move at constant speed. We are having this electric field. Yeah, we will fill one with the slow trailing electrolyte mixed with the samples, and other fill with the fast leading electrolyte. So L, and this is your S1, S2, that is your slow trailing, and the mixed with the sample S1 and S2, and the L1 are the fast leading. Then electroosmotic flow will start. Sample lines over speed, so they will go and mix together somehow. We will see this S1, S2 be in middle, in between your T and L, and then your S1 will be separated, S2 will be separated. On the left mainly your T will be there, on the right your L will be there. The sample continues to accumulate if the sample concentration approaches the concentration of leading electrolyte, and sample segregate into the discrete zones. So once faster moving components separate completely from slow moving one, it creates a region of depleted charges between the two that increases the resistance and therefore local voltage in that region. So this increased voltage causes and the slower component to migrate faster and close the gap, thereby concentrating it, increasing the conductivity of the zone until it matches that of faster ion. Just a second. Then comes your, I think the last one, this is very important, that is immunoelectrophoresis. These are the antibodies that are produced by immune system in response to foreign micromolecules. So each antibody binds specifically to one feature on one micromolecule, and this allows the use of antibodies for detection and quantitation of specific proteins in complex mixture. So one electrical potential is applied to study of antigen-antibody reaction. It is called immunoelectrophoresis. The antibodies are electrophoretically separated and antigens diffuse towards each other, resulting in the precipitin arcs. So here we have these unpaired arcs over the antibody, and the antigens are attracted towards these arcs. So when we run these electrophoresis, as a result, precipitin arcs will be formed due to the antibody and antigen complex formation. So in this method is carried around the 2% agar gel medium. An antigen mixture is applied into the small circular wells and cut out of agar, and the initial electrophoretic separation is carried out depending on the charge and the molecular weight. So main advantage that the spreading of band is minimized due to the application of light field, and pH gradient high resolution could be achieved. But here we have to have ampholites, high relative concentration, high voltage power source, and needs in the vicinity of 2 to 50 volt. And this we have discussed yesterday in detail, isoelectric focusing by that professor from our from BioRed. So I will not continue this part. This is already done. All the methods, how things were happening. Yeah, this two-dimensional, after the isoelectric focusing, we did this two-dimensional electrophoresis. Yeah, so yeah, that's it from this perspective. So I will share these things after the lectures are over. So for the moment, I think we have to make this. We still have 30 minutes. So mostly we do this thing on for a day, but we can continue with our next part. So I will also share here this AGAROSE electrophoresis protocol for DNA. That how you do this for the DNA and practically. So these protocols, students, whenever you want to work in your lab, so try to make your protocol of your own. So you can have this protocol, you can do your things, but you know, once you will be doing them on a regular basis, you will do some changes to it. So you might be not doing the same protocol. There will be something you will modify with the time. So try to write in on your own words and yeah, make your own lab book out of it. So I will continue in the same series with the blotting techniques. So it will make sense to continue the electrophoresis techniques that we have done, and the remaining 4 techniques, that is your southern, northern, and western, and southern western, northern western, zoo blotting and dot blotting. So always remember this snow drop, S for southern, N for northern, W for western, and D for DNA, R for RNA, P for protein. So southern is meant for to analyze DNA, northern is done for to analyze RNA, and W is done to analyze your protein. Yeah, O is just to remember. So we will talk about southern blotting, northern blotting, western blotting, and southern western blotting, northern western blotting, dot blotting, and zoo blotting. So southern blot hybridization detects target. Just a second, student. ये चले जाए, आप लेके गए हैं पहले? आप लेके गए हैं कैंडी को पहले? वो कुछ लोगों तो नहीं कहीं काटेगी ये? अच्छा, ठीक है। बस इसे कोई गंदी जगह मत सूंघने देना। बस फटाफट वहाँ पे जो है ऑटो मार्केट है, वहाँ से बस घुमा के ले आना। नहीं तो मैं 9 बजे ले जाऊँगा इसको, क्लास खत्म हो गए। चलो ठीक है। पर उठा के लेके जाना पड़ेगा इसको। 1 मिनट बच्चों, जस्ट अ सेकेंड। इसको नीचे तक ना उठा के लेके जाना पड़ेगा, नीचे जाती नहीं है। उठा लोगे आप इसे गोदी में? चलो कैंडी करके आओ। इसको खाना पड़ेगा। Okay, so southern blot, it was discovered by E.M. Southern in 1975. They have to identify DNA fragments by size fraction by the gel electrophoresis. So in this property, we label our single-stranded DNA with radio label probe and identify what we are looking for, complementary sequence or target sequence according to the design of the probe. So in this procedure, once you run your gel electrophoresis with your DNA sample, so your nucleic acid will show various bands, and we need to, this is a radio label markers for specific sizes, and this is your electrophoretic gel. So after running the gel, your all DNA samples will be separated, and you can put over the buffer and make a stack of paper towels on both and transfer over the nitrocellulose membrane like we have discussing from last 2 or 3 days. And then after the transferring, you get your nitrocellulose membrane to be out, and then you put with the particular radioactively labeled probe acid, and then you watch these over the hybridized nucleic acid. So whether a particular gene is present, how many copies are present in that organisms, we can identify that. We can identify gene mapping, genetic pattern, mutation, various cancers like sickle cell mutation, paternity testing is possible, personal identification, and lots of more applied field. So let's see how the southern blotting is done in practically. I think I don't have videos for that. So okay, I play this video will not have any sound, but you can have a look over it. There will be some, yeah, subtitles will be coming how this southern blot technique is done in the lab. So this will be a virtual lab for the same. Yeah, so enjoy this video about southern blotting and practically how you do it. I'll just come in a second. I think there is some, is it lagging going on or? Hmm. So I think I can stop it here only, or shall we continue? It's not usual as it should go, I guess. So what I'm speaking, it seems to be coming quite late. This always happens whenever I play video. I don't know why. What's the reason behind it? Yeah, I know that. I know that. But don't you worry. I will share all these PPTs